Hello everyone, my name is Cindy and today I will be presenting cellular signaling IP topic number 1. So there are 4 forms of signaling mechanisms: autocrine, paracrine, endocrine, and justacrine signaling. In autocrine signaling, the signals are usually produced by the cell itself for the cell. The signal doesn't really go anywhere, and the signal activates any particular mechanisms that need to happen in the cell. In paracrine signaling, the signal is released locally and then carried to nearby target tissue. This tends to happen with growth factors. In endocrine signaling, a signal is released from a faraway source and then is carried to target cells via the circulatory system. A great example of this are hormones, where they are produced in one organ and then carried to various different areas of the body. Finally, juxtacrine signaling, you can have a signal that is released from a cell, but its target cell has to be making physical contact with that cell. So they are only able to signal with cells in their local areas. In this slide, I provided a figure from our book that gives you a good idea of how each of these types of mechanisms work and how they can interact with the other cells. Here I'm going to explain to you the proposed model of signaling transduction. Basically, you get a ligand, which is a molecule that can start or stop a cellular process depending on its presence, that binds to a receptor. These receptors have a binding site that is specific for ligands. Once the ligand and receptor bind together, they activate a signal transduction, which in turn relays signals in the inside of the cells. Once the signal transduction is started, there can be specific responses that are activated in the cell, or some sort of change in gene expression is activated, depending on the response from the signal. In this last slide, I'll be explaining some basic type of signaling pathways that are listed in our book. Before we get started on the different pathways, you should keep in mind that for the most part, these ligands are hydrophilic. They also have the ability to bind to specific transmembrane receptor proteins, depending on which pathway they are taking. First, we have ligand gate ion channels, which allow the regulated flow of selected ions across the plasma membrane. Pretty simple. Ligands bind to receptors and then allow certain ions to pump through the plasma membrane and into the cytosol of the cell. Second, we have G-protein coupled receptor, or GPCR. The ligand binds to the receptor that causes a change in conformation, which activates G-proteins. G-proteins are a type of GTP binding regulatory protein in the plasma membrane that mediates signal transduction pathways. Third, we have enzyme-coupled receptors, which, for example, a receptor kinase can bind to a ligand, stimulate it, and then transmit signals through the phosphorylation events in the cells. Finally, we have nuclear receptor pathways, which the hydrophobic ligand is coupled with a hydrophobic cytosolic receptor. It binds together and then moves into the nucleus of the cell and can regulate the transcription of genes in the cell. That has been my presentation today, and thank you so much for listening.